Biotechnology companies are engineering brilliant molecular concepts, but translating those concepts into approved therapies often stalls right at the point of operational execution. The friction happens at the clinic level. Sites become overwhelmed with administrative tasks, patients drop out because of grueling visit schedules, and data quality begins to degrade. Solving this requires a specific framework to bridge the gap between scientific ambition and clinical reality. We call it the clinical harmony equation. It relies on three specific components: radically simplifying your protocol design, deploying purposeful clinical-grade technology, and empowering your human site staff. Today, we will deconstruct the specific operational bottlenecks where trials fail and lay out a strategic blueprint to optimize digital endpoints. In modern drug development, flawless execution on the clinic floor carries the exact same weight as the viability of the molecule itself. The most common point of failure for site retention is an overburdening protocol. Asking too much of clinical investigators immediately puts a trial at a disadvantage. Principal investigators operate in a competitive environment. If your protocol requires excessive administrative overhead, they will simply route their patient population to a simpler, competing study. This animated funnel chart demonstrates attrition caused by a highly complex protocol. Participants abandon a study midway if the volume of tests causes exhaustion. Watch what happens when we streamline it. By removing friction, the retention rate remains high from start to finish. You can achieve this by intelligently grouping patient visits and consolidating data collection. Reduce the number of days a patient has to travel and minimize the time they spend in the clinic when they do. Apply a strict rule to your endpoint selection. Only include measurements that directly advance the drug to the next regulatory or scientific inflection point. Exclude everything else. Collecting massive amounts of secondary data is ultimately self-defeating if the complexity of gathering it causes the entire trial to stall. To reduce patient burden, there is a push across the industry to adopt cheap, off-the-shelf consumer gadgets under the assumption they automatically improve patient centricity. It is tempting to offer participants a standard $30 or $40 retail blood pressure cuff. It seems like an easy way to lower the barrier to entry and let patients collect data from home. We frequently see consumer wearables, like the smartwatch shown here, proposed for critical continuous monitoring tasks. These consumer devices lack the precise body positioning and validated algorithms required for rigorous clinical-grade analysis. They are built for general fitness tracking, not regulatory scrutiny. This chart compares the output of a generic smartwatch against a dedicated medical patch. True patient centricity requires dedicated clinical solutions that don't compromise data integrity with erratic noise. A dedicated patch ensures the correct sampling frequency, providing dense, uniform data you can confidently trust. The mandate here is uniformity. Trials must deploy a single hardware solution with real-time digital capture across all patients, eliminating the variability introduced by mixed personal devices and paper diaries. Sending patients home with consumer hardware might seem convenient, but it introduces analytical noise that destroys confidence in the data and directly threatens regulatory approval. Perfecting the technology stack only solves half the problem. The trial still requires a highly capable human element to keep it moving forward. Project managers serve as the operational lubricant in this process. They force disparate data systems, site locations, and scientific strategies to align. Project managers build rapport across the network. When studies inevitably hit logistical roadblocks, they provide the transparency, trust, and rapid problem-solving necessary to maintain momentum. On the clinic floor, the retention of participants comes down to the site nurses and clinical staff. Their ability to address concerns and provide basic human comfort ensures that patients persist through demanding study schedules. When this human support structure is missing, bottlenecks compound. A principal investigator might encounter a technical issue and spend 4 hours on a support call, completely derailing the trial timeline for that day. This donut chart visualizes the metric of success when a dedicated human quality management team intercepts site issues. With the right operational structure, you hit a 90% resolution rate within 15 minutes. The most advanced digital strategy in the world will collapse under its own weight if you fail to empower the human staff responsible for daily care and crisis execution. This human-first approach extends directly to how we integrate artificial intelligence into clinical data management. Right now, the safest and most effective use for AI is not autonomous decision-making. It is operational surveillance and rapid data quality enhancement. Take a look at this dataset matrix representing oncology CT scans. When uploads arrive from dozens of different sites, AI can instantly highlight a mismatched patient ID number before it enters the system. In this context, AI acts as a continuous quality assurance layer. It operates as a force multiplier for human experts, like radiologists, by significantly reducing their margin for error before they even begin their analysis. However, there is a strict strategic warning to observe. If an AI model is allowed to independently analyze primary endpoints without oversight, it risks hallucinating patterns within the clinical data. Biotechnology companies must implement rigid human governance. AI must be treated strictly as a companion tool that flags anomalies for human review, never as a replacement for scientific judgment. When tightly governed, artificial intelligence systematically reduces the noise in your dataset. This improved clarity can allow for smaller sample sizes and shorter trials, without sacrificing patient safety or analytical rigor. Every element of this execution framework, from simplifying endpoints to deploying clinical-grade hardware and governed AI, requires precise planning long before a trial begins. You must align your endpoint strategy and patient population targets with global regulatory authorities as early as Phase 1. A concrete example of this foresight is implementing a collect and hold strategy for cardiac safety data during early healthy volunteer trials. By capturing high-dose ECG data early and holding it in reserve, a sponsor can secure a regulatory waiver later on. This completely eliminates the need to fund a highly expensive QT interval study in Phase 3. But that strategy is entirely dependent on the decisions made on day 1. You cannot secure that Phase 3 waiver if you captured your Phase 1 data on unvalidated consumer-grade hardware. The clinical harmony equation maps a complete route from Phase 1 to market approval. It demands simple endpoints, purposeful technology, firmly governed artificial intelligence, and highly empowered clinical staff.